Greetings everyone and welcome to the partition data analysis tutorial. This is protocol number 2 in a 3-part series of protocols entitled Phylogenetic Inference using RevBayes, published in Current Protocols in Bioinformatics. Protocol number 1, which we've already done, is the substitution model tutorial, where we learned how to analyze an alignment assuming that the entire alignment is evolving under the same substitution model. In this protocol, we are going to learn how to analyze an alignment by assuming that different parts of the alignment evolve under different substitution models. So we might want to partition our alignment by gene or by codon position, and in doing this we are doing what we're calling a partition data analysis. And so in this protocol, we're going to learn how to specify and select among alternative data partition schemes in RevBayes. And in the first part of the tutorial, we're going to use a model where we assume that all sites are evolving under the same substitution model. So this is the same as the uniform protocol number 1 tutorial. In the second model, we're going to partition our data by gene and assume that each gene evolves according to the same process, but that the different genes have different substitution model parameters. And in the third model, we're going to partition the data by codon position and allow each different codon position to have its own substitution model parameters. And in the end, we're going to summarize each analysis using the maximum a posteriori tree to find the inferred phylogeny. And in an advanced exercise, we'll use Bayes factors to select across various partitioning schemes. We won't go into that in this tutorial. So all of the data files that you'll need you can find them over in the data files section along with all the scripts. We're going to be using 2 alignments of mitochondrial genes from primates. And just by using the source command in RevBayes, you can run each of these scripts and hopefully everything should work for you. For the overview and background, I'll leave that up to you to read through that. It's not very long, but I will skip ahead to the exercise section of the tutorial now. Welcome to the first exercise in the partition data analysis tutorial. In this exercise, we're going to be analyzing our data by assuming that all of the sites evolve under the same substitution process. So this is the same as what we've done in the previous substitution model tutorial. We're going to do a concatenated non-partitioned analysis. So first things first, we have to load in our data. We have 2 alignments. We have 1 alignment of COX2 genes from primates and 1 of cytochrome B. And so first we read our data from COX2 into a data object using the read discrete character data function, giving the file name in the data directory. And then we do the same thing for cytochrome B. And then because in this analysis we're going to be treating all of the data as a single alignment, we're going to combine the 2 data sets into one by using the RevBayes concatenate function. And so all you need to do there is give both of your alignment objects to the concatenate function, and it returns a workspace object called data in this case that is also an alignment, and if you examine it, you can see that it will have a number of characters equal to the sum of the number of characters in the 2 alignments. So we're going to take this concatenated data and use it later. We're also going to get some helper variables out of the data object. We need the number of taxa, so we're going to get a constant node with the number of taxa using the n-taxa function, or method, the n-taxa method of the data object. And then we're going to compute the number of branches in our tree, which is 2 times the number of taxa minus 3. And then we also need a list of the taxa in the data set which we can get using the taxa method of the data object. And then following other tutorials, we are going to start moves and monitors vectors using the vector moves and vector monitors functions. This just creates empty moves and monitors vectors for our analysis. And then on to the substitution model part of the analysis. So we're just going to assume that our entire alignment is evolving under a single GTR plus gamma model. And so we are going to specify this model just the same way that we did in the substitution model tutorial, where first we need a prior for the exchangeability rates of the GTR matrix. So we're going to specify that using a Dirichlet distribution. So we're going to create a exchangeability rates parameter distributed according to a Dirichlet distribution with a concentration parameter, which is just a vector of all ones. So this is just a uniform Dirichlet distribution across 6 parameters. We have 6 exchangeability rates. And then we're going to create some moves for that parameter. We're going to use a beta simplex move and give it a pretty strong concentration and lighter weight than the next move, which is a Dirichlet simplex move. We also need to specify a prior for the 4 stationary frequencies of our GTR matrix, which we're calling pi. And we'll do it the same way using a uniform Dirichlet prior, but over 4, a simplex of size 4. And that'll give us our prior on our base frequencies. And we'll apply moves to that parameter as well, using the same beta simplex and Dirichlet simplex moves, which we append into our moves vector. Finally, we can create our substitution matrix, the Q matrix, as a deterministic function of the exchangeability rates and the base frequencies using the FN GTR function, or GTR matrix function. And that's all we need to do for the Q matrix. So we're using a GTR plus gamma model, so we also have to specify prior on the shape parameter of our gamma distribution for rates across sites. And so we're going to call that alpha, and we're going to draw it from a uniform distribution from 0 to 100 million. And we're going to use that shape parameter to create a discretized gamma distribution with 4 rate categories, where each of the bins in that discretized distribution corresponds to the median of each bin. And so we're going to use the discretized gamma function to create a deterministic node of those rates, 1 for each rate category, that is a function of our shape parameter that we will be estimating. And we also need to create a move for the shape parameter alpha, and we're just going to use a scale move with lambda equal to 0.1, and we're going to append that into our moves vector. So we're also using a GTR plus gamma plus I model, which allows for some probability of certain sites to have a substitution rate equal to 0. And so we're going to specify that probability, and we're going to call it P invar, invar. We're going to estimate this probability by drawing it from a prior distribution, a uniform beta distribution. So here we create a stochastic node drawn from a beta 1,1 distribution. And then we create a move for that parameter, a beta probability move, which will operate on the p-invar move. And so we're going to use this parameter to estimate the fraction of sites that have substitution rate equal to 0, essentially. Finally, we need to create the prior for our tree topology. And so we're going to do that by assuming that our tree topology is drawn from a uniform distribution over topologies, with the constraint that we're going to specify an outgroup. And in this case, our outgroup just has a single taxon, so we still have the full uniform distribution over topologies. In this case, our outgroup is Galeopterus variegatus. And so we're going to create a clade object with that taxon name as the argument to the clade function, and create a workspace object outgroup, which is a clade object. And then we're going to use that outgroup as the argument for the outgroup parameter of our uniform topology distribution, which we also need to provide the list of taxa that we got from the data object earlier. And then we can create our stochastic node for the topology drawn from this uniform topology distribution. And then we're going to create moves on the topology, so we're going to use two different types of moves. We're going to use nearest neighbor interchange and subtree pruning and regrafting. And so we create an NNI move on the topology and an SPR move on the topology. And we give them weights according to the number of taxa. So if we have a bigger tree, we're going to propose these moves in proportion to basically the number of taxa. If we have a bigger tree, we're going to propose these moves more often because mixing on a bigger tree is harder than mixing on a smaller tree. And we append those moves into our moves vector. And then we need to create parameters and moves for our branch lengths of the tree. And so for that, we're going to make a for loop where we go from 1 to the number of branches, and for each I in that vector, we create a stochastic node and add it to this branch lengths vector, and draw each of those from an exponential distribution with rate parameter 10. So the mean of this distribution will be 1 over 10. And so the mean branch length should be 1 over 10, a priori. And for each branch length, we also create a scale move with the default weights and tuning parameters. And then because monitoring the branch lengths tends to be a little bit complicated in terms of its interpretation, we're going to create a deterministic node to monitor the tree length, which is just the sum of the branch lengths. And that'll make it easier for us to determine or assess the convergence among the branch lengths, because as we're rearranging the topology, the meaning of one branch in one topology will change when we go to a different topology. So monitoring a single branch length parameter doesn't give us the same meaning across the entire run of the MCMC, so we use the tree length, which has a consistent meaning across samples in the MCMC. Finally, we need to create our branch length tree object that we will actually use to compute the likelihood of our data. And so for that, we combine the topology and the branch lengths together using the tree assembly function to create a deterministic node called psi here that represents our branch length tree. Finally, we put it all together by taking our branch length tree, our substitution matrix, our gamma distributed rates across sites, our rate categories, our proportion of invariant sites, and take all of those and give them as arguments to our Phylo-CTMC function, which creates the stochastic node for our sequence data. And we need to tell it that we have type DNA. And then for our first analysis, we're going to be doing an unpartitioned analysis where we concatenated our data into this single data object. And so we just clamp our sequence node at the value of that data so we can compute the likelihood. Label that node as part of the likelihood and fix it to the value of that data. And so now we have all of the components of our model specified, and so we're going to wrap them all together in a model object using the model function, using any node from the DAG, and in this case we're going to use the substitution matrix. We're going to call it my model. So we need to specify some monitors for our MCMC. We can do this much like in previous tutorials. We're going to create a model monitor that monitors all of the parameters, all the stochastic parameters in the model. And we're going to monitor the tree topology separately, our branch length tree separately, by creating a file monitor with psi as the argument. And we're going to also create a screen monitor that we're going to use to monitor the gamma shape parameter and the proportion of the variant sites and the tree length, which we'll monitor less frequently than we do to the files. And then we'll initialize our MCMC just like in previous tutorials. In this case, we're going to have 2 runs, and we're going to combine those a posteriori using the mixed method. And then we're going to run our MCMC for 30,000 generations and tune every 200 generations. And then in the script, after that's completed, we're going to read in our tree trace that we created in our file monitor and summarize it to get our maximum a posteriori tree using the map tree function. And that's the end goal of this exercise, is that map tree. In the second partition data analysis exercise, we will be partitioning our data by gene region. So we had 2 genes in our data set, and we're going to create a substitution model now to analyze the evolution of each gene separately in a combined analysis. And so we need to, this time, create multiple substitution matrices and parameters for those matrices for each gene. And we'll get into how we do that. So first we're going to clear our workspace because we're going to redeclare some variables that using a different structure, using the same name as previously declared variables. So we need to just empty our workspace. And we're going to start by creating a vector of our data file names. Using this v vector function, we'll feed in the file names in the data directory, one for our COX2 gene alignment and one for our cytochrome B alignment. And then we'll get some helper variables along the way. One of them is the number of partitions. So in this case we have 2, but in the case that you had more files, you could handily get the number of partitions using just the size of this vector. And then we're going to read in our data, one for each file name, by looping over this file names vector from 1 to the number of files, and then read in each file name using the read discrete character data function and create a separate data object in this data vector for each file name. And then just as we did in the previous exercise, we'll get our helper variables. We'll get the number of taxa, the number of branches, and create our moves and monitors vectors, and so on and so forth. Now we need to specify the parameters of our GTR matrices for each partition. And in order to do this, we're going to have to loop over the number of partitions that we have. And so if we have a large number of partitions, it's somewhat cumbersome to manually specify the parameters for each one. So we're going to loop over the number of partitions and create a vector for each parameter. And so in the case of the exchangeability rates, we're going to create a vector for the hyperparameter of the Dirichlet prior distribution, where each element of this vector is itself a vector with 6 ones. So that's our concentration parameter for each Dirichlet distribution. And then we're going to give our Dirichlet prior that parameter and create a stochastic node for our exchangeability rates and add it to the vector. So this vector, this is a vector of Dirichlet distributed stochastic nodes. And then we'll append a move for each one of these to our moves vector using a beta simplex move. And then we'll do the same thing for the stationary frequencies, where in this case we'll just use a Dirichlet distribution of size 4. Our base frequencies are we're going to call pi, and we're going to add them to this vector, and then similarly create a beta simplex move for each one. And then we will have a similar loop where we go over each partition and create a separate GTR Q matrix for each partition by pulling these exchangeability rates and the base frequencies from the vectors we just created, and create a deterministic Q node for each matrix and add it to this vector Q. And then we do the same thing for our gamma distributed rates and for our proportion of invariant sites. We're basically copying the code from the previous exercise and putting it inside of a loop and adding everything to a vector. So again, we will have a separate shape parameter for each partition, drawn from the same prior as before. And then we have a separate set of deterministic rate categories for each partition that are drawn from this discretized gamma distribution using our alpha shape parameter. And then we have a proportion of invariant sites for each partition, and drawn from a uniform beta distribution. Now, because we're also doing this partitioned analysis, we'd like to assume that each partition can have its own substitution process, but also its own overall substitution rate. So what we're going to do is assume that partitions share branch lengths, but that each is scaled with respect to one another by a certain partition rate multiplier. And so we're going to draw our partition rate multipliers from a Dirichlet distribution, and then in the end we're going to multiply that rate by the number of data sets, which ensures that the average substitution rate across partitions is equal to 1. And so what we're going to do is draw that partition rate multiplier vector from a Dirichlet distribution, where the concentration parameter is just a vector of 1s of length equal to the number of partitions that we have. And so we have a simplex here of size equal to the number of partitions, and we add some moves for that simplex, similarly to the way we did for the other Dirichlet distributed parameters. And then we get our partition rates for each partition by multiplying this vector by the number of data sets. And that'll give us a deterministic node with the partition rate multiplier for each partition. So this is a vector, a deterministic vector. Finally, we need to create our tree topology prior distribution, which we're actually going to do in exactly the same way as in the uniform partitioning exercise. So nothing here has changed, actually. And we're going to put it all together by creating a vector now of our sequence stochastic nodes, one for each partition, and we're going to give each one our branch length tree parameter that is shared across partitions, and then feed in our substitution matrix, our Gamma rates are proportional to various sites, and then here we're going to specify that we're using a branch rate multiplier using this branch rates argument, and we're going to give it the partition rate multiplier that we computed earlier. And then we're going to clamp that sequence data at the partition that we stored in our data vector earlier. And then going forward, the rest of the exercise is exactly the same as the partition uniform exercise, so I'll leave that up to you to set that up. Just remember to use different output file names so you don't overwrite the files that you created in the first exercise. I'll see you in the next exercise. So in the third exercise of the partition data analysis tutorial, we're going to be partitioning our data by codon position and by gene. So in the first exercise we didn't have any partitioning scheme. In the second exercise we partitioned our data by gene, and now we're going to partition also by codon position. In order to do that, we will be copying our data partitions into another vector called data, and then setting the codon partition of those partitions using the set codon partition function of the alignment object. So we'll create our first codon partition for our COX2 alignment by copying that alignment into the first element of a data vector, and then calling the set codon partition function on that alignment that we copied and giving it a vector of codon positions. And so in this case we're going to partition the first two codon positions separately from the third codon position, and so we give it a vector of 1 and 2, saying that this first partition is the first and second codon position partition for COX2. Then we assign the third codon position for COX2 by copying the data again into the second element of the data vector and setting the codon partition to 3. So you'll notice that we're copying our data. We still have an original copy of the alignment that we're copying into a new vector and then changing the codon partition of those copies. Then we repeat the same thing for cytochrome B, making sure that we increase the element of our data vector index, increase the index of our data vector. So 3 and 4 is going to be cytochrome B, 1 and 2 is COX2. But we do the same thing, first and second codon positions for cytochrome B and third position for cytochrome B in the fourth partition. And what we end up with is 4 data partitions, 2 for each data set. And so in all of this we've assumed that each alignment is in frame, meaning that each codon in the nucleotide alignment is aligned, it's aligned by codon. So you'll need to make sure that your data is aligned by codon in order to use this type of partitioning. And then everything else for specifying the substitution models is the same as in the previous exercise. We're going to loop over each partition and create a different GTR plus gamma plus I matrix for each one, and then a different PhyloCTMC node for each partition as well. It's just that in this case, instead of the partitions containing data from each gene, now the partitions contain data from different codon positions. And the rest is the same. But don't forget to rename your output files so you don't overwrite the previously created files. Finally, we have some wrap-up exercise questions for you to consider after you've run each partition data analysis. So first of all, you can review the posterior estimates of each analysis's parameters. So remember that data subsets 1 and 2 are for COX2 in the codon position partition analysis, and 3 and 4 are for cytochrome B. And aside from the tree topology and branch links, we have different parameters for each partition, so you can compare the parameter estimates between each analysis and between each partition. For example, the clock rate is about an order of magnitude faster for codon positions 3 than it is for first and second codon positions. And you might think about why that would be, and then identify some other parameter subset relationships that you can compare between different partitions and between different models. And so, for example, one question you might consider is, do you think that partitioning by codon position or by gene has greater influence over the site's evolutionary mode? So, in other words, is it more important to partition by codon position or by gene? And you can get an idea of that by looking at how much information you get about the different parameters for those different partitions, and whether they are very different between partitions or whether they're similar. The second exercise question for you to look at is comparing the maximum a posteriori trees that you get from each analysis. And so we've got 3 inferred map trees that you can look at in Figtree. And if you enable node labels and click display posterior from the menu, then you can look at the posterior probabilities of the different clades in the different trees, and you can compare that between different models. For example, are the more complex partitioning models providing higher or lower clade support? And why do you think that might be? Finally, in order to estimate which partitioning scheme is actually fitting the data better, we can use a Bayes factor analysis to compare the fit of different partitioning schemes. And so you can see the model selection using Bayes factors tutorial for more details on this. But in brief, you can do this analysis by modifying your analysis scripts to conduct a stepping stone or path sampling analysis. And the way you do that is first by giving your model to a power posterior function, the power posterior function in RevBayes, using a number of categories that kind of creates a path from the prior to the posterior using a certain number of steps. And in this case, we're going to use 127 steps. And so we'll have this power posterior that we will then burn in and run for a certain number of generations. And after that's done, we can use our stepping stone sampler function using the output from our power posterior analysis to estimate the marginal likelihood of each partitioning scheme. And so after we've created our stepping stone sampler, then we compute the marginal likelihood using the marginal method of that object. And then again, using the path sampler, it's exactly the same, except instead we're using this path sampler function, but you estimate the marginal likelihood using the same function name. And so then you can obtain estimates of the marginal likelihoods for each partitioning scheme, and then use those to compute support for one partitioning scheme over another in the form of a Bayes factor. And for more information on that, again, you can see the model selection using Bayes factors tutorial.